The urea cycle is a very important cycle in the human body since it is responsible for conversion of ammonia into urea. And as you can remember very well, we said ammonia is very toxic. Ammonia is very toxic. So if there are high levels of ammonia in the blood, you would experience the following symptoms. One, coma, hand tremors, changes in consciousness, excessive sleepiness, and confusion and disorientation. Okay, so what are some of the causes of high ammonia levels in the blood? Okay, number one, it can be the liver disease, decreased blood flow to the liver, genetic diseases of the urea cycle, hemolytic disease of the newborn. You can have a kidney failure being the source of high ammonia levels in the blood. And also, if you have hepatic encephalopathy, can also be the source of ammonia problem in the body. So today, let's focus our discussion on the urea cycle. And we said the source of urea cycle is the breakdown of proteins in the body. And another source that we had mentioned when we were discussing about the structure and anatomy and function of the stomach is the reaction whereby the urease enzyme that is produced by the chief cells of the stomach acts upon urea and produces ammonia. And we say that ammonia is transported through blood to the liver, whereby it's converted to urea. And then after that, it's transported to the kidneys where it is excreted. Now, the urea cycle steps happens in the liver hepatocytes. I mean, the process of the urea cycle happens in the hepatocytes. So the first steps happen in the mitochondria of the liver hepatocytes, and the last stages happens in the cytoplasm of the liver hepatocytes. Hepatocytes are simply liver cells. Hepatocytes are the liver cells. So, in the first step, I like to say that ammonia and carbon monoxide are converted into carbonyl phosphates. Ammonia and carbon monoxide are converted into carbonyl phosphate, and the enzyme responsible for this reaction is known as carbonyl phosphate synthase 1. Carbonyl phosphate synthase 1. In this reaction, two ATP molecules are used, and these two ATP molecules are the source of the phosphate in the carbonyl phosphate. Now, ammonia becomes the source of the first amine group of urea. Ammonia becomes the source of the first amine group of urea. Now, the carbonyl phosphate synthase 1 needs an activator, and that activator is called N-acetyl glutamate. N-acetyl glutamate. And N-acetyl glutamate is produced when acetyl-CoA and glutamate are reacted in the presence of N-acetyl glutamate synthase. So the enzyme that's responsible for conversion of N-acetyl-CoA in glutamate into N-acetyl glutamate is N-acetyl glutamate synthase. Okay, now in the first step, now this carbamyl phosphate enters the urea cycle. The carbamyl phosphate that is produced when carbon monoxide and ammonia are converted by an enzyme called carbamyl phosphate synthase. It enters the urea cycle and combines with ornithine. It combines with ornithine. And remember, we said ornithine is an unprotein amino acid. Ornithine is an unprotein amino acid. So I've said carbamyl phosphate, it enters urea cycle and it combines with ornithine to form citrulline in the presence of an enzyme called ornithine transcarbamylase. Ornithine transcarbamylase. I would like also to mention that citrulline is also an non-protein amino acid. Okay, here, so we have two non-protein amino acids, that is ornithine and citrulline. So in this step, we have said carbamyl phosphate is combined with ornithine to form citrulline. And now this citrulline that is formed is transported out of the mitochondria into the cytoplasm of the hepatocytes by an enzyme called ornithine translocase. So the enzyme that transports citrulline from mitochondria into the cytoplasm is called ornithine translocase. Okay, now after forming our citrulline and it has been transported into the cytoplasm, now this citrulline is converted with aspartate and undergoes a condensation reaction to form adeninosuccinate. So the condensation reaction between citrulline and aspartate forms adeninosuccinate in the presence of an enzyme called adeninosuccinate synthase. So the enzyme here that converts citrulline and aspartate through a condensation reaction is called adeninosuccinate synthase. Here the aspartate becomes the second source of the second ammonia of the urea. So we said that the first source of amine group in urea was ammonia, and now we have said the second one is we have said is aspartate. Okay. So this reaction also utilizes another ATP molecule. So far we have 3 ATP molecules that have been used. So the third step, adeninosuccinate that we have produced by combining citrulline and aspartate through a condensation reaction, and we said in the presence of an enzyme called adeninosuccinate synthetase. So the adeninosuccinate is cleaved off to form adenine and fumarate. So adeninosuccinate is cleaved off to form fumarate and adenine in the presence of an enzyme called adeninosuccinate lyase. Now the fumarate is used in the production of NADH in the TCA cycle, and the adenine moves forward in the next step of the urea cycle. So we have said adeninosuccinate is cleaved into arginine and fumarate, and fumarate is transported to the TCA cycle to form NADH, and the arginine moves to the next step of this urea cycle. So in the last step, now this arginine undergoes an hydrolysis reaction to yield urea and ornithine. So the last step is arginine being hydrolyzed into urea and ornithine in the presence of an enzyme called arginase. The enzyme here is arginase. Ornithine is transported back to the mitochondria. Remember that this reaction is happening in the cytoplasm. So ornithine is transported back into the mitochondria and is used in the second step, whereby we said ornithine is in the second step, whereby we said ornithine combines with carbonyl phosphate to form citrulline. You can remember that. So ornithine is transported back into the mitochondria to form citrulline. Okay. So the overall reaction is just the one that is shown here. We started whereby we had ammonia combining with carbon monoxide, and then aspartate, and then we said aspartate combining with citrulline to form adeninosuccinate, and then we said 3 ATP molecules were used. And after all this reaction that we are seeing here, the end result is urea has been formed. So that is the urea cycle. And in the next tutorials, I will discuss about the regulation of the urea cycle and the disorders that are associated with the urea cycle. So we'll discuss about the disorders of the urea cycle and the regulation of the urea cycle. So very quickly, so let us go through what we have learned today. We have said carbon monoxide combines with what? We have said carbon monoxide combines with ammonia to form carbonyl phosphate in the presence of an enzyme called carbonyl phosphate synthase 1. And we have said in this reaction, 2 ATP molecules are used. And then we said ammonia is the source of the first amine group of urea. And then we said carbonyl phosphate synthase 1 requires an activator which is known as N-acyl glutamate. N-acetyl glutamate. And we said N-acetyl glutamate is formed by a reaction between glutamate and acetyl-CoA in the presence of an enzyme called N-acetyl glutamate synthase. Now, again, we said carbonyl phosphate that has been produced enters the urea cycle and combines with ornithine to form citrulline. So, in the presence of an enzyme that we call ornithine transcarbamylase. Now, the citrulline that is formed is transported from the mitochondria into the cytoplasm, whereby other reactions continues. And the enzyme that we said that is transporting citrulline from the mitochondria to the cytoplasm is called ornithine translocase. Okay, so the citrulline that has been formed now combines with aspartate in the condensation reaction to form adeninosuccinate in the presence of an enzyme called adeninosuccinate synthetase. Now here aspartate becomes the second source of the second amine group of the urea. Now this step also consumes another ATP molecule. You get that? Now, The next stage, we said that the arginosuccinate is cleaved into arginine and fumarate. And now we say that fumarate is used in the production of NADH in the TCA cycle, while now our arginine moves to the second step of the urea cycle. Now this the last stage of the urea cycle is whereby arginine undergoes an hydrolysis to yield ornithine and urea. Now urea now is transported into the kidneys whereby it's excreted, while the ornithine joins back the cycle and it combines with citrulline. No, it combines with carboxyl phosphate to form citrulline. And now citrulline again is transported towards the cytoplasm and the reaction continues, the reaction process continues. Now this reaction is a cycle because you see now at the last step we have formed ornithine and now ornithine has gone back and then the reaction again will start again all over again. So that's all. And now I'll have to say bye.